It's so nice to see everybody. And while I say hello, I'm going to do the hardest part of any Zoom meeting and try to share my screen. So we're going to talk a little bit about news from the front, what's going on in the research world. We're going to talk about what we know and what we don't know and why it's so important that Curals is funding the people who are getting to those answers. And we're going to talk about what you can do to help yourself and those you love while we go on searching for the answers in Alzheimer's disease. So some of you will have seen this slide before. I always like to start with this because it's a reminder that it wasn't that long ago that we thought of aging as kind of a just, it was what it was. And if you just were not feeling well or your body didn't feel good or your brain started to go, you'd go to your doctor and they'd just kind of say, well, gosh, you're 65. That's the end of that. It's just all downhill from here. Now, while it's true that we cannot reverse aging at this point and we cannot stop Alzheimer's disease, it is not true that we have to assume that everybody is going to lose their cognitive function as they age. We have wonderful examples of resilient aging people. Sometimes we like to call them super agers, who, when we test their cognitive function at 90, 95, 100, they test like they're 55 or 60. We have lots of us who are typical agers who do see that things like processing speed slow down as we get older, and maybe it's a little harder to remember where we left our keys or the name of the person we sat next to in first grade. But overall, we are still functioning just fine in our day-to-day lives. And then on the other hand, unfortunately, most of us who come to Cure Alzheimer's Fund events have experienced with a loved one what dementia looks like, what mild cognitive impairment, which is the stage when people are still relatively functional, but they are starting to have impairments in their executive function and their memory that are causing them issues. And in dementia, where people have truly become impaired because of the loss of cognitive function and memory. What we want for everybody is at least typical aging. What we really want to achieve, what we aspire to, is resilient aging. But we believe, and science is telling us, that we do not have to accept mild cognitive impairment or dementia. So, how did we get here? Alzheimer's disease is not actually just originated in 1906 when the first clinical case was presented in a case history. In fact, the word dementia I could trace back to the year 600. I believe it was BC, not AD. And it was essentially loss of self is the meaning of the original term. So we've known about dementia for as long as we've had people able to get old. And even in fact, some young people get dementia for various reasons, although it's much less common. But the first case of Alzheimer's disease was characterized in 1906. Interestingly enough, Augusta Dieter, who's the woman pictured here, who was the patient, was actually an unusual Alzheimer's patient because she was only in her early 50s, which would make her a early onset patient. Most Alzheimer's patients do not develop any symptoms of any real impact until they're 65 or older. But after she died, Dr. Alzheimer and Dr. Fisher, who were the clinicians and scientists who studied her, looked into her brain and they saw the same pathology that we are still studying today. They saw tau tangles inside of neurons, and they saw beta amyloid plaques around neurons. They also saw lipid droplets in glial cells, which are part of the innate immune system of the brain. So the fundamentals of Alzheimer's disease could be identified even with the very, very, what we would think of as primitive tools that were available to people back in 1906. But that's how we know that she, in fact, had Alzheimer's disease and not another driver of dementia. And we'll talk more about that in a minute. The great news is that even though we have these wonderful drawings from the microscopes that were able to look at the pathology in 1906, we now have far better tools. And I just have some example images on the right here to show you what these look like. We can now see into the brain of a living person as their pathology begins to accumulate. That has told us an enormous amount about how this disease actually works. When they looked at Augusta Dieter's brain, they didn't know whether the beta amyloid plaques came first or the tau tangles came first. And we are able to see that accumulate now in a living brain and place things in time. We have PET imaging, which is essentially how we do see this in the brain. That's the colorful images at the top. And we also have MRIs that can show us as people lose brain volume through neurodegeneration. That's the loss of tissues and cells that is caused by the accumulation of Alzheimer's pathology. We are able to look at the human genome. We've mapped almost in its entirety. We always talk about how it's absolutely been mapped all the way. It's not quite the case, but it's pretty close. We can look at blood vessels and see materials passing along them and where there are leaks that make a difference in the blood-brain barrier that protects the brain. And we can even get all the way down to cellular resolution. So this image all the way on the right shows microglia coming towards a neuron because they've been attracted by issues in that neuron that they want to come do their housekeeping function in. It's really extraordinary what we can see, and that is how we are making the kind of progress that we are making at this point. So I said that in order to be Alzheimer's disease and not another driver of dementia, you have to have three characteristic pathologies. The first is beta amyloid plaques, which form around neurons. Beta amyloid is primarily produced in the brain by neurons, but it actually is then trafficked to the cell membrane, and or the amyloid precursor protein is trafficked to the cell membrane, where it is cut in a very set number of steps. And if the cuts happen in the right order and at the right places, you end up with a beta amyloid peptide. That's just basically a nice fancy word for a small protein. That peptide gets released into the space among neurons, where after it finds other peptides like it, if it is the right length, if it is a beta amyloid peptide of 40 or 42 length, it is prone to basically snap together like a Lego and form larger and larger clumps until you end up with an insoluble beta amyloid plaque. That's what we generally talk about most with Alzheimer's pathology. People talk about whether they have plaques in their brains or not because it's so associated with Alzheimer's disease and not with any other diseases. There are other things called amyloids, but beta amyloid plaques are specific to Alzheimer's disease. In Alzheimer's, we start to develop these plaques 10 to 15 or even 20 years before the development of symptoms. And in a process that we are just beginning to understand, and we have a number of researchers looking at this, the development of that beta amyloid plaque, as we see more and more of them in the hippocampus and other brain areas, eventually begins to induce the development of something called neurofibrillary tau tangles, which I cannot say quickly. NFTs are tangles of a protein called tau that exists in your neurons as part of the skeletal support system that allows your neurons to move materials from the center of the neuron, the nucleus, down to the axons and synapses. Synapses are the connection points with other neurons. Neurons need to be able to transmit electrical and chemical signals across those synapses in order to communicate. That circuitry is the fundamental basis of cognition and memory. So when we have disruptions to those synapses, we then see the circuitry start to fail. And when we see disruptions to the circuitry, when we see material accumulating around the neurons that isn't supposed to be and within the neurons that isn't supposed to be, you end up with an immune response where your brain's innate immune cells say, wait a minute, that looks like trash, or worse yet, that looks like an invader. Our job is to come in and clean that up. And if we get really, really determined that this is bad stuff, that this is some sort of invader or a disease, we're going to be extremely aggressive in cleaning up. And we're going to kill off everything around what we see to try to essentially quarantine the damage. The process by which those immune cells gather around debris and pathology is called neuroinflammation. And neuroinflammation can lead to the loss of both diseased cells and debris and to the loss of healthy cells and material and synapses. And that's when you end up with neurodegeneration. As you can see in the picture all the way on the right, the brain of a healthy senior looks much, much bigger and thicker almost than the brain of somebody who is late in Alzheimer's disease. Unless you have amyloid, tau, and neurodegeneration, you do not actually have Alzheimer's disease. And what's fascinating is there are people who can live with some aspects of these pathologies, but they don't get that neurodegeneration. And then they don't seem to get the cognitive symptoms. We call these folks resilient. And we have a lot of people who are spending a lot of time trying to understand what drives resilience. But it seems that the neuroinflammatory response in these people is much more modulated. It turns on and it turns up, but then it turns itself back down. So again, you have to have all three of these to actually have Alzheimer's disease. You can get dementia, which is the clinical presentation of cognitive decline and cognitive impairment from other causes. But if you have these three, if we can see that in your brain, or we can find that out looking at your cerebrospinal fluid or in your plasma, your blood, looking at your biomarkers, then we will know that you actually have Alzheimer's disease. So genetics drives familial Alzheimer's disease, the early onset that we most often see kind of dramatized in movies and things like that. Still Alice, for example, was a movie about somebody who had early onset familial Alzheimer's disease. It's actually exceedingly rare. It's something like 1 to 3% of all cases. It's the result of mutations in just 3 genes, all of which relate to the production of beta amyloid. The fact that these genes are almost entirely fully penetrant, and by the way, we thought they were entirely penetrant till about 2 years ago, is one of the reasons we know that amyloid is so central to this disease. Down syndrome folks also are at extraordinarily high risk of Alzheimer's disease because they have a third copy of the chromosome on which the precursor protein, that amyloid precursor protein, sits. So they also overproduce beta amyloid. For these folks, the symptoms are going to develop generally before 65, sometimes as young as 30 years old. The pathology may be developing as young as 20 years old. We do now have just a handful of cases where we are seeing people who seem to be resilient to these genetic variants. The only way we have discovered this is because we have vastly expanded our genetic monitoring. It's fantastic to discover that because it means there's hope for families that we had never been able to give hope to before. And in all cases, it seems to be the fact that they have counterbalancing genetic variants that may actually change the impact of what we would have thought of as guaranteed genetic variants. But again, these cases are very rare. Then we have another group of extremely rare cases, about 1 to 3% of all cases. This is again a group that is very newly recognized. These are people who would not necessarily even have been identified as Alzheimer's patients until we were actually able to look inside living brains. For amyloid, we started to be able to do that around 2005. For tau, we started to be able to do that around 2012, 2013. These are people who develop Alzheimer's disease as early as their 50s sometimes. They don't seem to have family history at all. They don't seem to have any obvious epilepsy or TBI or other forms of trauma that might trigger an earlier onset of Alzheimer's disease. Our best hypotheses right now is that they have spontaneous mutations. So rather than inheriting a mutation in a gene that leads to Alzheimer's disease, they've just unfortunately experienced a spontaneous mutation. But again, we are just starting to see these people and recognize them for who they are because we are doing so much more sort of monitoring than we used to. And then finally, we have sporadic Alzheimer's disease, which is a very terrible description for what this is. This doesn't mean sporadic in the sometimes it's there and sometimes it's not case, although Alzheimer's disease can be experienced that way by a patient. What this means is it's not predicted by somebody's family history and genetic background, or not entirely predicted. Many people with sporadic late onset classic Alzheimer's disease do have family histories. They clearly have genetic backgrounds that increase their risk or lower the age at which they might develop Alzheimer's disease. But there are no gene variants that guarantee sporadic Alzheimer's disease. APOE4 is the genetic risk factor that we most associate with sporadic Alzheimer's disease. That is the biggest risk factor for people of European and Asian descent, but we're now discovering that it may not have the same impact in people of descent from certain areas in Africa. That's actually really, really exciting because if we can understand why it doesn't have that impact in those folks, we might be able to decrease the impact in everybody else. There are many, many known risk genes now for sporadic Alzheimer's disease, many genes that we can say increase or decrease your risk of developing this or change the likely age of onset. That's actually powerful because it means that every one of the pathways in which those genes sit is a potential pathway for intervention. So it makes everything more complicated, but in a disease as complicated as Alzheimer's, we want to have as many opportunities to intervene as we can. For the genes that are known contributors to sporadic Alzheimer's disease, the ones that are most common, meaning more people carry them, generally affect the immune response to Alzheimer's. So again, talking about that neuroinflammation piece that's so important. The rarer ones are have to do with neuronal survival, their ability to withstand pathology like beta amyloid and tau. So what we know about Alzheimer's now that we can see into living brains and we can study people for years and years and years, particularly folks that we know because of their family history and their genetic variants are going to develop the disease, is that people develop the pathology decades before they are going to develop the symptoms. So there's a very, very long period of time where people are beginning to overproduce and accumulate beta amyloid pathology. We believe in the folks who have the familial gene variants, that the issue there is that they are overproducing beta amyloid. We believe, and these are hypotheses, that for folks who are developing sporadic late onset classic Alzheimer's disease, they are increasing the production of beta amyloid as they age, but the bigger issue is that their brains are no longer able to clear it out as well as they used to. Their debris and housekeeping mechanisms are not successful in maintaining an appropriate balance. But it does mean that we have this very long period of time where if we could stop the production of beta amyloid or cause it to be cleared more effectively, we might stop all of the downstream effects that it has. The new drugs that have been approved that I'll talk about a little bit in a minute are very much aimed at this period of time before you have significant symptoms and significant tau pathology. What's interesting is that you see the development of tau pathology really kick in right around the time that you start to see the development of symptoms. So that's that orange line, that's the second one there. We think about secondary prevention as could we stop the development of tau pathology? In particular, could we stop its path through the brain? It spreads through the brain, it propagates through the brain in a very specific pattern that is essentially specific to Alzheimer's disease. There are other primary tauopathies where tau is the main form of pathology. It's not induced by something like beta amyloid. But in all of these cases, we believe if we could target tau effectively, maybe even generically, maybe it wouldn't have to be the specific form of tau seen in the different tauopathies, we might be able to stop a lot of the neurodegeneration and the cognitive impairment. Recent discoveries by labs that we fund, in particular the Holtzman lab at WashU, has shown us that in fact there is a very strong secondary immune response of the peripheral adaptive immune system to the development of tau pathology that is very closely aligned with substantial neurodegeneration. That is fascinating, exciting, and kind of out of nowhere because until very recently, we didn't believe the adaptive immune cells from outside the body got into the brain absent a catastrophic infection. So as people start to develop that tau pathology, they start to develop symptoms and they move into that mild cognitive impairment stage. That stage is one in which many people have very high quality of life still. They are bothered by their symptoms. Their loved ones may recognize them. People who interact with them on a casual basis may not even recognize that they are having issues. If we could keep people in that stage longer, we could give them substantial quality of life for a much longer time than we can now. The drugs that we have, that's their best bet, essentially, is to keep people at that stage longer, at least for the use that that they're approved for the time being. But then finally, the tau pathology starts to affect neurodegeneration and brain structure, and then we start to see really significant symptoms, significant memory dysfunction, and then finally significant clinical dysfunction. Often the spectrum starts with executive function, where people stop being able to plan very well. It's hard to follow a recipe and figure out what steps to do in what order. That executive function isn't always accompanied by significant memory decline. But once we see memory decline with the executive function, it gets harder and harder for people to go about their day. And finally, as the memory dysfunction and the executive function really are lost, you start to see people who really are not able to care for themselves anymore. Often along the way, you see significant emotional upset, and it's actually agitation in Alzheimer's patients that is the most significant reason their loved ones need to put them into institutional care. There is a new approved treatment for agitation in Alzheimer's disease patients, and that's fantastic because we really know how hard that is for caregivers to handle. But ideally, we would do something about at the treatment stage where we would be able to stop people from developing the full clinical dysfunction, including emotional upset. So as I said, we do have some first drugs, and they are big news because they are our first FDA-approved disease-modifying drugs, but they're really limited. So these drugs are designed to task your innate immune system in your brain to remove amyloid pathology once it has begun to accumulate. They're only approved for people who already have significant amyloid pathology and some memory concerns. And we have found in clinical trials that they do not do very much, if anything, for people who already have significant tau pathology. So there's a very limited window of time in which patients need to start these drugs if they're going to see any benefit. The drugs are able to slow progression by about 25 to 30% during the course of the clinical trials, which only lasted about 2 years. But they did not stop or reverse disease progression. So the benefits are not that substantial. Now, if we can keep somebody at a stage where they're still able to drive and function and play cards with their loved ones and enjoy their families, and they get to stay in that stage for a good bit longer, that's really promising and it's really exciting to a lot of people. But these drugs do have a significant risk of side effects, especially for APOE4 carriers and people with certain comorbidities related to their blood vessel health. And so some doctors do not believe that the risk-benefit ratio is favorable, and they do not want to prescribe them. There are lots of patients who do want to take them. There are lots of people who were in the clinical trials and who are on these drugs and believe they are seeing real benefit. We don't know anything beyond what the clinical trials can tell us because the data just doesn't exist yet. But there is some suggestion from the data that the slowing of progression may extend further and be more than what we saw in the trial. We won't know that till we know that, though. The challenge is that there's a lot of steps that are required before somebody can access the drug, and we don't have nearly enough clinicians who are actually available to people to bring them in to get them all the testing that they need, like MRIs to make sure that they don't already have brain lesions. Then they need regular MRIs to make sure the drugs are not harming their brains. They need to be tested to make sure they have amyloid in the first place. There's a whole wealth or a whole breadth of testing that has to be done both before the drugs are prescribed and afterwards. And then the drugs are currently given to people through infusion, which requires sitting in an infusion chair once or twice a month for about an hour. So access to these drugs is very, very challenging. They are also extraordinarily expensive to the system. They're going to become cheaper as they become subcutaneous injections, and we anticipate that development in the next 18 months or so. But this cannot be our answer. This approach does not do enough, and it doesn't reach enough patients. So it's exciting because it has shown us that amyloid is, in fact, a promising target, but this cannot be the be-all and end-all of what we offer to patients. So we've had a lot of progress, and we're really proud of the key contributions of Cural's funded scientists. But as I just said, the progress is not nearly enough. We do have more and better biomarkers. That's a way of testing things, for example, in your blood to tell us about what's going on in your brain and what your future looks like. It's incredibly important to have those be biomarkers that we can access easily. And there is now a clinically or a number of different clinically approved blood tests that can be ordered to assess somebody's risk of Alzheimer's disease and to be a differential diagnosis if somebody has memory concerns for whether what they are experiencing is Alzheimer's or isn't. As I said, we have these two FDA-approved drugs. We've determined that the innate and adaptive immune system response is central to this disease. It's central to whether you actually develop the neurodegeneration that's going to damage your cognition versus you just end up with a whole lot of pathology, which may make your brain work not quite as well, but might not be devastating. If we can figure out how to modulate that, we might be able to make everybody resilient to the pathology. That resilience is biological and clinical. We want to figure out how we can also help people who do have the pathology and have developed some clinical symptoms to not go on to develop greater degree of Alzheimer's disease. And we have all kinds of tools that allow us to look into brains and actually design and test drugs faster and cheaper, both in cell cultures, in animal models, and in people. It's not enough. But we are moving faster and faster than we ever did before. The progress in the last 10 years dwarfs what we did in the 10 years before that. And it felt like we didn't make much progress at all in the 10 years before that. So it is really exciting, but there is just so much that we need to know still. For Cure Alzheimer's Fund, that means that we need to make sure we are finding actionable ideas. We are always looking for knowledge that will actually benefit the entire field and accelerate progress across the entire field. We have the privilege of being a not-for-profit. We don't have to make money by anything that we do. That is extraordinarily important because a lot of the knowledge that we need that is so important, why and how amyloid induces tau pathology, that's not something that you're going to be able to get a patent on, nor should it be, because if one entity patents it, only that entity is going to have any ability to actually pursue and get to an answer that can benefit everybody. And it will be done at a cost that nobody else will be able to afford. So we fund an intentionally diverse research portfolio. We want to fund all kinds of great ideas because it's going to be a jigsaw puzzle. There are going to be bits and pieces from all kinds of different areas of neuro research, immunology research, and research from other disease areas that need to be put together to solve this incredibly complex disease. We want to be the test bed for great people to explore these ideas. We want to have a fast fail model where if you have a great idea and a track record of being right, we empower you to go test that as fast as you can and to figure out whether it's a yes or it's a no. We're not going to punish you if it's a no, if it was a great idea. We're just going to make sure as many people learn about it as possible so we don't have to keep chasing that rabbit. We can go after the other ones. We produce seed funding so that the data that comes out of our projects can be leverageable into NIH grants and for-profit investments by other entities downstream. Again, we want to accelerate the entire field. We want an answer for Alzheimer's patients. We're not necessarily going to get credit for it. We're certainly not going to make money off of it. That's okay. As long as Alzheimer's patients are benefited, Cure Alzheimer's Fund has succeeded. And we require our scientists to cooperate, not just compete. Yes, they compete. They're a really competitive group of people, but we want them to compete for the best ideas and to work with one another to refine their ideas. And they do that. They love our meetings. They come to our meetings when they don't want to travel anymore. They still show up at ours because they have the opportunity to talk to the best in the field and make sure that they are learning as fast as everybody else is so that they can in turn produce the best possible projects. That world-class research leadership group that we've convened are our decision makers. They do actually receive funding, and some of the best work that we fund comes through them, but they also help us identify other talent out there, and they tell us what is hot and important and next in Alzheimer's disease so that we are always ahead of the curve. We try to be great partners to them, and right now that is more important than ever. The research environment, the scientific environment, particularly in the United States, is under attack. We know that there is cultural mistrust of science right now. It is heartbreaking. We also know that there is a real change at the federal level in the level of support that the federal government seems intent on providing to our scientists. If we don't feed the scientific entity, if we don't make sure that great new ideas receive funding, then nothing happens downstream except the more Me Too drugs that are based on drugs we already have. We will not end up with new approaches, new targets, new cures. We want to make sure that great ideas don't have to wait. Operationally, this is really hard on us. It's a lot easier if you have set funding deadlines and you tell everybody exactly what you're going to fund in a given year and you don't look at anything else. We don't do it that way. We want to fund your great idea as fast as you have it. We're going to try to get you the money in weeks, not months, much less the years that it can take for NIH funding. And we want you to tell us, or we want our scientists to tell us what matters most, not for us to direct them. So as I said, we are trying to feed the entire Alzheimer's ecosystem. That means that most of our research is early stage. It's actually earlier than somebody would be able to get intellectual property for, and it's certainly earlier than somebody would be able to get biotech or VC funding for. We will support projects when we identify them as great ideas early in wet lab stage. We will help them get over what we call the valley of death, where for-profit funders want more and more data and de-risking, and not-for-profit funders often don't go far enough. So we will help bridge that valley of death. But we are thrilled when we fund research that will be published, but isn't actually going to be necessarily patentable, because it means that somebody out there that we may not fund and we might not know can build on the great idea that we allowed to get out there. And that may lead to a cure for Alzheimer's disease. And that to us would be fantastic. So we fund all kinds of different great hypotheses. We don't limit ourselves to just one. It's interesting, sometimes we fund hypotheses that are a little bit in opposition to one another, because that's how great science works. You have to let ideas compete with one another. We obviously cut things off when we have data that disproves a hypothesis, but we also celebrate when hypotheses are supported and need refinement as new data is brought to bear on them. We also fund not just the responsive projects where somebody wakes up in the middle of the night and says, but what if? We also do convene big consortia that attack some of the hardest problems in Alzheimer's disease that are thorny long-term projects that really benefit from a number of labs going after them together. We require these consortia to actually cross institutions. A lot of academic and research institutions want all of the money to stay within their walls. We won't fund a consortium that way. They actually have to go across. And the consortium we're working on putting together this year, which is a stretch goal if our budget can reach it, will probably have 6 labs in at least in 3 different continents. So these consortia really do seek the best and the brightest wherever they may be, and really, really force, which they actually enjoy, but their institutions may not celebrate. We force them to cooperate and collaborate across those institutional walls. The immune response is clearly at the frontier of Alzheimer's research. About a quarter to a third of our portfolio every year right now is going into projects that are looking at the response of the immune system in Alzheimer's and how we might be able to modulate it. It's going to be quite complicated because Alzheimer's disease is not caused by a single germ. This isn't like I caught COVID and there's this invasive pathogen in my body, and if I could just do something about it, this would all go away. Alzheimer's disease is a disease in which natural cells, systems, and processes go awry. And so we can't overreact to our own self, or else we're going to have an autoimmune disorder or an autoimmune cytokine storm that's going to be very damaging. Instead, we need to help our immune system turn itself up to the right level and then turn itself back down. So it's going to be very complicated, but it's also very promising, because therapies that can rely on our own tools, our innate systems to fight our battles for us, are much safer over the long haul than therapies like, say, chemotherapy for cancer, where we're essentially hoping to kill as many cancer cells as possible before we kill too many of our own cells. So again, that's that modulation of the immune system, but we just don't have the deep enough understanding yet to know exactly how to modulate it and which cells we want to modulate. Just a few years ago, I would have been focusing this conversation on astrocytes and microglia. I still say and would say that microglia and astrocytes are incredibly important to this conversation, but now we're talking a lot about CD8 positive T cells. They were not a part of the Alzheimer's conversation 5 years ago, and we now have a number of labs that pioneered the work that identified their importance and are already figuring out how we might be able to task them to actually help us fight this disease, much as we see CAR T therapy in cancer. CAR T, the T is for T cell. In Alzheimer's disease, we're excited and funding a new project that's trying to determine whether there's a way to have something called a CAR M therapy, where we would look at macrophages that would take on microglial functions. This is like science fiction science, to be quite honest. Nobody would have thought of this or talked about it at all as recently as last year. So early stage research is the key to filling the drug development pipeline. I've told you a lot about things that we're working on where we've got unanswered questions and we're still trying to understand some really basic pathways and processes. But there are a lot of therapeutics in clinical trials, including phase 3 clinical trials. A phase 3 clinical trial is sort of the last stage before potential FDA consideration for approval and marketing to the public. We've got 37 different therapeutics in phase 3 clinical trials right now. A number of them, of course, are Me Too drugs, meaning that they are versions of Kisunla and Leqembi that are the drugs that are already approved, versions that are, for example, subcutaneous injections instead of being infusion therapeutics. So 37 sounds like a good number. It's not good enough. Not enough of these are truly new approaches. And a number of them, we don't know what will happen with them, but they're not all going to be successful. On the other hand, we do have a lot more targets in clinical trials than we used to. For a long time, all we had were the anti-amyloid immunotherapies. Leqembi and Kisunla are the successful ones of those. But we now have targets like the development of tau, the propagation of tau, different anti-inflammatory approaches. We just have a whole lot of different things that are being targeted. Some of them will need refinement. A lot of the work that we do goes into figuring out how those could be refined if they don't succeed, or how they can be improved if they do succeed. So we have a sort of a feedback loop with what's already in clinical trials. So this is really our call to action this year. Thanks to our donor community, we did have a record year in 2024. $30 million of research in 1 year was phenomenal. The year before I started at Cure-Alz, this is my 10th year at Cure Alzheimer's Fund, we funded right around $6 million that year. So to hit 30 just 10 years later is extraordinary. But we won't be able to maintain that level unless we actually have a stretch fundraising year this year. So that is something that we are really excited about trying to do. It's the message that we're trying to get out into the world. We also want people to be aware of the challenges that the scientific community are going to face if federal funding takes the enormous cut that has been talked about in just the last few days. It is very scary to imagine what the scientific enterprise will look like if the federal level of support declines the way it has been proposed. We have the best scientific infrastructure in the world right now. The NIH is the largest funder of biomedical research in the world. It feeds the rest of the world, but it also protects American leadership in science. We intend to do everything we can to support our scientists and to protect them from this, but there is no way our funding alone can match up with what the NIH has provided. So really excited about what our scientists are doing, really determined to empower them to keep doing it, but this is a very scary time for Alzheimer's science. Getting to how we can protect ourselves. This is an excerpt from the 2024 report of the Lancet Commission on Dementia Prevention. So the chart that you see is really about dementia, not specifically Alzheimer's, but many of the risk factors for dementia in general are also risk factors for specifically Alzheimer's disease. You'll see on this list on the left that many of these are not modifiable, but some of them are. Our choices during our life do matter. In particular, exercise and movement have extraordinary data supporting that they can change outcomes. We don't know how to prevent any one person's case of Alzheimer's, and anybody who tells you that they know how to do that right now is probably trying to sell you something. On the other hand, we do know that at a population level, people who exercise more, who don't smoke, who protect their cardiovascular and metabolic health, who eat a healthier diet. These are people who are less likely to ever develop Alzheimer's disease. And if they do develop it, they are likely to have many more healthy years before they do so. So these choices really matter, and they are choices that each of us is able to make. Other choices are not up to us, but as a society, we can do something about them. Air pollution is a significant driver of dementia. We worry a lot about social engagement, which is something that we need to do more about as a society, not just as individuals, although that's a choice each of us can try to make as well. And there's a lot about education that matters. When you control for almost everything else, including genetics, more years of education continue to be protective across every single study we have ever looked at. So it's really important that we actually invest in our children. So how do we shield our brains? Many of you will have seen this. This is courtesy of Dr. Rudy Tanzi, the chair of our research leadership group. I like this because it's very simple. There's a lot more we can talk about, and you saw a lot more in the slide before this, but this is pretty easy to keep in our minds. We need to sleep. It's really hard to sleep. Alzheimer's pathology disrupts sleep. Disrupted sleep can contribute to the development and propagation of Alzheimer's pathology. So sleep is a really tough one, but everybody can practice better sleep hygiene. We need to handle stress. It's amazing to see what a difference this can make. Meditation has actually proven clinical benefits, but it's not just meditation, it's however you personally do better to manage stress. For me, it's exercise, which is already on here too. That does not mean that I am out there doing Ironmans, not at all. Exercise is really about moving more and moving more frequently, not just about the number of steps you get in a day, or did I run for an hour continuously before I then sat in a chair for 11 straight hours. We really want people to be interacting with the world, especially with one another. There are parts of your brain that only get active when you interact with another person. It's actually even different interacting in person than it is through a screen. And we want people to interact with the world because it's important to keep your body moving in space. So one of the things that's quite interesting is your vestibular system is how your body knows where it is in space. And it's one of our oldest systems. It only gets engaged if you are actually moving in different ways, not just in a straight line and certainly not sitting still. It's really important to learn new things. That's not doing brain games. It's actually things as different as Spanish. If you never spoke Spanish before, sitting in your car and thinking, I need to go to the grocery store. I'm going to sit here and in my mental map plan a different way to get there than I usually do. Learning to brush your teeth with your left hand. I have to tell you, I brushed my nose many times before I succeeded on that one. Every time we force our brain to do something different, to learn something different, we encourage it to support its own plasticity. Our brains can still develop new connections and even neurons throughout our lifespan. And then finally, diet. People love to talk about, well, is it this supplement? Is it that supplement? Far more important and with far better data than any supplement is getting a lot of fiber in your diet to feed your gut microbiome, eating less meat or red meat, and a lot less processed food. If you can't tell what it was when it first was a food, it's probably too processed and you should reconsider eating it. That's one rule of thumb. The other is if you look at the list of ingredients and it's more than 5 and there are too many you can't pronounce, it's probably too processed and you should make a different choice. The other one is vaccines. There is emerging evidence, epidemiological and mechanistic, that's showing us that people who get more vaccines for all sorts of different diseases have a lowered risk of a dementia diagnosis in the 5 to 10 years after that. We don't have a great explanation for this yet, but since we know vaccines are good for us for other reasons, this is one of those, do the thing your doctor is already telling you to do, because it's also going to be good for your brain.